We're going to continue talking about renal physiology, and we're going to focus primarily on the proximal convoluted tubule. And from a conceptual standpoint, we can see that the proximal convoluted tubule receives the ultrafiltrate that is passed through the glomerulus and has created the ultrafiltrate from the plasma that enters into the Bowman's capsule, and the creation of ultrafiltrate, which contains many different electrolytes, anions, cations, as well as basic constituents such as glucose and amino acids. So in the proximal convoluted tubule, there's a series of reabsorption from the lumen of the proximal convoluted tubule into the capillary system, and eventually goes to become the systemic circulation. Likewise, a secretion from the capillary lumen into the lumen of the tubule, of the proximal convoluted tubule, which will eventually be secreted via the urine as an elimination of certain toxins and metabolites. So like we said just now, the proximal convoluted tubule can be thought of as reabsorbing many different electrolytes, anions, and cations, and the secretion of certain organic anions and cations such as urate, certain keto acids, and the secretion of certain cations such as creatinine. So we're going to look in further detail of the proximal convoluted tubule. So if we say there's a filtrate from the plasma that enters into the Bowman's capsule, the blood flow, which is the plasma, gets filtered through a capillary system, which is the glomerulus, and passes through, depending on the filtration process. There's an ultrafiltrate that's produced that contains many different ions. And we can think of the proximal convoluted tubule as reabsorbing approximately 75% of this entire filtrate, this entire ultrafiltrate that was created by the glomerulus. The proximal convoluted tubule reabsorbs approximately 75% back into the capillary system, which eventually becomes systemic circulation once again. And we can think of the proximal convoluted tubule as the primary function of reabsorbing sodium. And in fact, about 65 to 75% of the sodium that's filtered through the glomerulus is reabsorbed back into the capillary vessel by passing through the proximal convoluted tubule cells and eventually into the capillary system. And the way that this takes place is actively transported using the sodium-potassium ATPase. And this implies that this is an active process that requires the use of energy. And that energy is in the form of ATP. And if we take a closer look, we can see that this active transport of sodium that transports 3 sodiums out of the proximal convoluted tubule cell into the capillary system, which is a blood vessel, for every 2 potassium that are secreted from the vessel into the tubular cell. So this sodium-potassium ATPase allows for the concentration gradient of the high concentration of sodium within the lumen of the proximal convoluted tubule to allow sodium to enter into the proximal convoluted tubule cell. And because of that use of active transport, or the use of energy in the form of ATP, this pumps out 3 sodiums for every 2 potassiums that enter into the proximal convoluted tubule cell. And the importance of this, first of all, is considered the reabsorption of sodium back into the systemic circulation. And the second important factor is because of 3 positive charges entering in to the capillary system, or thought of as leaving the tubular cell, for every 2 potassiums that are entering, there's a net negative positive charge leaving the cell or a relatively negative charge within the tubular cell. So therefore, this relatively negative charge within this proximal tubular cell creates the driving force for other positive ions or cations to enter into the tubular cell. And those cations can be thought of as potassium, calcium, and magnesium. So this involves the reabsorption of these certain cations from the ultrafiltrate that was produced through the filtration process of the Bowman's capsule. So we're able to reabsorb the sodium through the process of active transport using the sodium-potassium ATPase. Because of the relatively negative charge within the cell, this is the driving force for the other cations such as potassium, calcium, and magnesium to be reabsorbed eventually back into the capillary system, which will lead to systemic circulation. Chloride is absorbed in two ways. One way is through passive reabsorption through the concentration gradient. It's able to pass through the tight junctions within the cell. And secondly, it's able to be reabsorbed through a co-transport system using the sodium. And likewise, be reabsorbed into the capillary system using the co-transport of the chloride and the potassium co-transport channel. Next, we're going to look at the counter-transport. Here we just looked at the sodium-potassium ATPase. We looked at a co-transport, for example, involving chloride, where two ions are moving in the same direction. Now the counter-transport is when the sodium positive charge is entering into the cell from the proximal tubular lumen and allows the secretion of the hydrogen ion, the proton, to enter into the tubular lumen. And the whole purpose of this function is to ultimately reabsorb bicarb, which has been filtered through the Bowman's capsule, the capillary system, which is a glomerulus. Bicarb was filtered and now part of the ultrafiltrate, and this bicarb would have continued to have passed through the convoluted tubule, down the loop of Henle, through the tubular systems to eventually be secreted via the urine. But because of this counter-transport system using sodium and hydrogen ion, this allows for the reabsorption of bicarb because this forms a carbonic acid, readily dissociates into water and carbon dioxide, which can pass through the cell membrane of the tubular cell, and therefore with the use of carbonic anhydrase can form the carbonic acid once again and allow for the dissociation, and therefore the bicarb can then be reabsorbed from the proximal convoluted tubular cell back into the capillary system, and therefore bicarb has been reabsorbed and can be used as the buffer within the acid-base system, which we'll talk about later, all in efforts to maintain an adequate pH so all the enzymes can work through the process of metabolism. And here we can see that as the bicarb is being reabsorbed, the proton, the H+, once again is being secreted through the process of sodium entering and the proton exiting, and the whole series of bicarb reabsorption continues. So as a recap, we can say that the proximal convoluted tubule reabsorbs the sodium, which we talked about, the sodium-potassium ATPase. Chloride is reabsorbed either passively or actively with the cotransport. Water readily is reabsorbed. The bicarb is reabsorbed, as we talked about before, through the secretion or counter-transport of sodium with the sodium and the proton allows for the reabsorption of bicarb, and a certain glucose, protein, and amino acids that are negatively charged also are drawn in through reabsorption. And lastly, we can say that the cations, such as potassium, magnesium, and calcium, are reabsorbed through the relatively negative charge that's created through this sodium-potassium ATPase. Now, next lecture we'll talk about the loop of Henle and the functions of the loop of Henle.